Imagine a world where scientists can rewrite entire chromosomes with surgical precision, transforming the very blueprint of life. A groundbreaking study led by Professor Gao Kaixia and her team at China's Chinese Academy of Sciences has just turned this vision into reality. Their creation, Programmable Chromosome Engineering (PCE) systems, represents a leap forward in genetic engineering, offering tools to manipulate DNA at scales previously thought impossible. Published in Cell on August 4th, this research doesn't just tweak genes, it reshapes the rules of genetic design itself. But here's where it gets tricky. The very tools that enabled this breakthrough were once held back by flaws so deep they stifled progress for years. Let's dive into how this team cracked the code, and why it's sparking debates in labs and living rooms alike. At the heart of the revolution is the Cre-Lox system, a workhorse of genetic engineering since the 1980s.
It's like a molecular pair of scissors and glue, cutting DNA at specific sites and rejoining it. But this system had three major roadblocks. First, the symmetry of lock sites meant edits could sometimes reverse themselves, undoing years of work. Second, the Cre enzyme's complex structure made it hard to tweak for better performance. Third, leftover LOX markers after edits created unpredictable genetic scars. These issues weren't just technical hurdles. They were roadblocks to curing diseases, engineering climate-resilient crops, or even understanding evolution itself. The team's solution? A triple-pronged attack. They first redesigned lock sites to be asymmetrical, like a lock and key that only fit one way. This reduced reversal errors by over 10 times, making edits near flawless. Next, they weaponized AI with their ICE-REC method, training algorithms to optimize the Cre enzyme structure. The result? A hyper-efficient Cre variant that outperforms the original by 3.5 times. Finally, they tackled the scar problem by combining their edits with prime editors, tools that can erase leftover lock sites like a genetic eraser. The outcome? A seamless, scarless genome. Putting it all together, the PCE and Re-PCE platforms now let scientists insert, delete, or rearrange DNA segments from thousands to millions of base pairs. For context, that's like editing a novel from a single sentence to an entire chapter. In rice, they created herbicide-resistant strains by flipping a 315,000 base pair segment. Proof that this isn't just theory, but a tool for real-world problems like food security. Yet, this is the part most people miss: the ethical tightrope. Can we trust such precision in human hands? What happens when improving crops means altering ecosystems we barely understand? And here's a provocative question to chew on. If we can edit chromosomes flawlessly, should we? The comments below are open. Weigh in on whether this is a miracle or a minefield.